In 2001, a geneticist at University College London took DNA samples from Jewish communities across 14 countries, communities that had been separated for over 1,000 years, living under different governments, speaking different languages, marrying into different surrounding populations, and compared their genetic profiles, expecting to find exactly what happens to every other population that separates for that long. Populations that separate for a millennium diverge genetically as a rule. They pick up local DNA, they lose the older markers. The genetic signal of a common origin fades over time and gets replaced by the signature of wherever those people ended up living. And that pattern holds across virtually every human population group ever studied at comparable scale with comparable separation periods. The Jewish communities did not follow that pattern, and the degree to which they did not follow it was not marginal or debatable, but was strong enough that the shared genetic signal across Ashkenazi Jews from Germany and Poland, Sephardic Jews from Morocco and Turkey, Mizrahi Jews from Iraq and Iran, the Beta Israel of Ethiopia, the Bene Israel of India, and the Mountain Jews of the Caucasus pointed clearly toward a common ancestral population that had lived together in a specific geographic region long before any of those communities formed. The signal pointed to the Levant, to the ancient Near East, to the region that modern maps call Israel, Palestine, Jordan, Lebanon, and Syria. And when the statistical models tried to date the point at which all of these geographically scattered communities last shared that common ancestor, the number they kept landing on was not medieval, was not ancient in the conventional sense, was older than anyone running the studies had expected. The standard model that geneticists were working with going into the early DNA studies predicted a mixed picture. A founding population in the ancient Levant that experienced dispersals starting with the Babylonian exile in the 6th century before the Common Era, and the Roman destruction of Jerusalem in 70 of the Common Era, followed by each diaspora community gradually drifting away from the ancestral profile as they absorbed local DNA from surrounding populations across centuries of separation. The early genetic data in the 1990s seemed to support this picture superficially, because Jewish populations did show elevated levels of genetic markers associated with Middle Eastern ancestry and did show some signature of surrounding populations, but the proportions were wrong, in a direction that the model could not cleanly explain, because the Middle Eastern signal was far too strong, and the surrounding population signal was far too weak for communities that had supposedly been absorbing local DNA for between 500 and 2,000 years. A 1997 study led by Michael Hammer at the University of Arizona analyzed Y chromosome haplotypes from Jewish and non-Jewish Middle Eastern populations and found a strong shared signal across the Jewish communities, with the Y chromosome pointing toward a common paternal ancestry rooted in the ancient Levant that showed remarkably little evidence of European or Central Asian admixture in communities that have been living in Europe and Central Asia for over a millennium. Then the mitochondrial DNA studies came in, and the picture became more complicated, because while the Y chromosome in Ashkenazi men with clearly Middle Eastern in origin, the mitochondrial DNA of Ashkenazi women showed strong evidence of European ancestry, specifically pointing towards Southern and Central Europe, a significant discrepancy that the data supported, explaining as Jewish men moving through Europe during early dispersals and frequently marrying local women who converted or were otherwise integrated into the community. The age question is where the research went somewhere that surprised even the researchers running the studies, because the tools geneticists use to date ancestral populations, including linkage disequilibrium, which measures how many generations have passed since a founding event by tracking the rate at which genetic variants get shuffled and recombined across generations, kept producing numbers that did not align with what the historical model expected. A 2010 study in the American Journal of Human Genetics, led by Bahar and colleagues, performed a comprehensive genome-wide analysis of 14 Jewish populations and compared them to a broad array of non-Jewish Middle Eastern, European, and African populations. And the core shared component, the signal that transcended the differences between Ashkenazi and Sephardic and Mizrahi and other communities, traced back to a founding population in the ancient Levant with statistical dating placing it conservatively between 2,000 and 3,000 years ago, with confidence intervals extending significantly further back. Later studies using newer methodologies pushed the dating in a direction that surprised the research teams, with a 2022 analysis published in Current Biology using whole genome sequencing on dozens of ancient DNA samples from the Levant, alongside modern Jewish population genomes, finding that the genetic profile of Jewish communities today most closely matches ancient populations that lived in the southern Levant during the Bronze Age, not the Iron Age, where the historical record documents the kingdoms of Israel and Judah, but the Bronze Age, the period before 1200 before the Common Era. The genetic signal was not pointing to the Iron Age kingdoms as the founding source, it was pointing to something older, and this finding carries enormous implications for how we understand the origins of the Jewish people as a genetic population, because the standard historical model treats the formation of the Israelite identity as a process that occurred primarily in the Iron Age, making the Bronze Age connection not an ancestral footnote, but a direct founding relationship. The ancient DNA evidence from archaeological sites across Israel, Palestine, Lebanon, and Jordan over the past decade has made this finding concrete by giving researchers the ability to compare modern Jewish population genomes directly against sequenced DNA from Bronze Age and Iron Age human remains, recovered from dozens of excavated sites, producing a picture of what the ancient southern Levantine population actually looked like genetically, with enough specificity to make the comparisons meaningful. What the ancient DNA shows is a population that was already distinct from other Near Eastern populations by the Bronze Age, carrying its own specific combination of ancestral components, including a significant contribution from the Neolithic farming populations that had spread into the Levant from Anatolia around 10,000 years ago, combined with older components from the Natufian culture that had inhabited the region since the end of the Ice Age. The Natufians are the critical piece of this story because they were a hunter-gatherer culture that lived in the Levant from roughly 15,000 to 11,500 years ago and are considered among the key ancestors of the farming cultures that later dominated the region. And when the Natufian genome was first published in 2016 using ancient DNA from a site in northern Israel, the researchers were able to compare it directly to the modern populations that showed the strongest genetic affinity. The modern population with the highest proportion of Natufian-related ancestry among living humans was not any Arab population from the surrounding region, was not the Druze or the Palestinians or the Bedouin, though all of these showed some affinity. It was the Jewish population. And this finding was noted in the original paper, and then largely passed over in mainstream science coverage despite the fact that it extends the Jewish genetic signal back through the Natufian lineage to populations living in the Levant at the end of the last ice age, 12,000 or more years ago. The work of Yosef Lazaridis and his collaborators at Harvard's Ancient DNA Laboratory has provided some of the most detailed resolution available on these questions by sequencing large numbers of ancient genomes from across the Near East and comparing them to both modern and ancient populations with the statistical precision that large sample sizes and whole genome sequencing make possible in ways that the early blood group and Y chromosome studies could not achieve. What this work has repeatedly found is that the genetic component that most distinguishes Jewish populations from other world populations is not a recent addition or a product of the medieval dispersals, but is a component that traces through Bronze Age Levantine populations and through those populations back to the Neolithic farmers who moved into the region and to the Natufian hunter-gatherers who preceded them, meaning the thread is not broken across the enormous time span it is being traced through. The distinction between a direct, unbroken lineage and a genetic thread that persists through mixing and population movement is the one that population geneticists spend considerable effort making clearly, because the Natufian connection does not mean that Jewish communities are the literal direct descendants of Ice Age hunter-gatherers in any simple genealogical sense. It means that the specific combination of ancestral components that distinguishes them carries a signal that was already present in populations living in the Levant before agriculture existed. The implication is that what genetic analysis keeps finding across studies from different research teams using different methodologies across 3 decades of increasingly sophisticated tools is a population whose founding genetic signal is older than the religious and cultural identity that formed around it, older than the kingdoms that the historical texts document, older than the Bronze Age civilization whose collapse restructured the ancient Near East, reaching back to the specific Levantine lineage that was already ancient when the first written records were being made anywhere on Earth. The coherence of the signal across geographically scattered communities is the finding that keeps requiring explanation, because a standard expectation for any population that separates and disperses across 14 different countries for over 1,000 years is significant divergence, and the Jewish communities show the opposite of significant divergence. They show a level of genetic coherence that is scientifically anomalous by the standards of what other human populations demonstrate under comparable conditions of geographic separation. The Y chromosome data and the whole genome data converge on the same geographic source, with the same time depth, the ancient Levant, with the dating consistently placing the founding population further back than the historical model predicted, and the ancient DNA comparisons consistently identifying the Bronze Age Levantine populations as the closest ancient genetic relatives of modern Jewish communities, regardless of which contemporary Jewish community is being analyzed. 3 decades of increasingly precise genetic research, beginning with blood group comparisons in the early 1990s and arriving at whole genome sequencing of dozens of ancient DNA samples in 2022, has produced a consistent picture that the Jewish people as a genetic population carry a founding signal that is older than anyone expected when the first studies were designed, rooted in a specific geographic region, preserved across 2 millennia of geographic dispersal through cultural practice, and traceable through Bronze Age and Neolithic ancestors to hunter-gatherer populations that were living in the Levant when the last ice age was ending, and the world that produced all subsequent civilization had not yet begun to form.